Good morning. Well, thank you very much for showing up that early on the third day. You're a very special crowd. What we would like to discuss today is, overall combination therapies again. But I think we want to dig a little bit deeper. I think we have heard a lot about the overall excitement about combination therapies and a lot of optimism. I think we'll try to be a little edgy and ask some of the questions. What does that really mean? I mean, how does all of this really translate into the daily practice? I have a fantastic panel here, and what I would like to ask them is to introduce themselves, and I would like to combine it with the introduction question. What do you really think is suddenly driving the proliferation of combination therapies? Ken, why don't you start off?
Thanks very much, and it's an honor to be here. I'm Ken Anderson from Dana-Farber, and I've been there for 36 years, and I work on multiple myeloma. And in the theme of combination therapies, we've been very blessed in myeloma. There's been a wonderful collaboration of biotech and industry with clinical and basic researchers and the regulators, the FDA, foundations, and most importantly, the patients. But we've had, in the last 12 years, we've had 16 new FDA-approved treatments, and last year we had 7 in 2015. As a consequence, patients are living 3 to 4 times longer. And in the theme of today of combination therapies, many of those drugs were approved as single agents. So the proteasome inhibitor that David Schenkein and we worked on together, for example, is a mainstay. The immunomodulatory drug, thalidomide, lenalidomide, and pomalidomide are also a mainstay. But very quickly, we and others have learned in genetics of the tumor, but also in the microenvironment in myeloma that, and honestly, no one agent, no one class of agents is enough. Combinations are really usually more than additive or synergistic. So we've combined the proteasome inhibitors and the immunomodulatory drugs. That's a standard of practice today. Pretty much universal responses, and half of the patients are getting molecular CRs. So we have genetic heterogeneity, multiple clones, multiple mutations right from the start, and combinations are used. We'll talk about this later in the panel, but I would just like to mention that it's not just combination of targeted. As we get into the immune therapies later, we're very excited about the immune therapies, not any one of them as a single agent, but combinations. A vaccine or an antibody in combination with something to prolong that response. We have exciting data with vaccines together with IMiDs or checkpoint inhibitors or even HDAC inhibitors that you can get a response against cancer, and you might even be able to have a memory immune response. So it's going to be combinations of agents to try to deal with the genetic heterogeneity, and in the immune area, combinations of agents to hopefully have a memory immune response and be able to deal with all of the mechanisms that are used to underlie relapse. So combinations, I always talk about teams. I won't talk about Boston or New York teams here. But combinations of agents, targeted immune and probably targeted and immune are in the future.
Excuse me. My name is Jeff Engelman. I'm a physician scientist at Mass General Hospital, where I direct the Thoracic Oncology program and Molecular Therapeutics program. I've been interested in combinations really mostly from a targeted therapy perspective in a few different veins. One is that in lung cancer, we know that certain targeted therapies can have a really dramatic effect on cancers, but they do not lead to cures. And a big part of our research is to understand how cancers become resistant. And often we find that there are specific mechanisms at play, and combinations can potentially overcome this resistance and kill resistant clones. So we've looked at combinations to overcome resistance and combinations to use up front, hopefully to prevent resistance. And one of the issues I'm sure we'll talk about is that we would love to use 4 or 5 drugs at once, but the therapeutic index for using many medications at one time is lacking with the current dosing and scheduling strategies. The other vein in which we use combinations is for cancers where there is no effective single agent therapy, and sometimes a combination might be effective. I think one of the situations that's clearest is BRAF mutant colorectal cancer, where BRAF inhibitors alone really aren't effective. But because of the signaling pathways, if you combine them with an RTK inhibitor, you can have much increased efficacy. And so it takes two drugs that actually either one alone in that cancer had minimal efficacy, but together can have actually real efficacy. And that's an approach now being used in terms of combinations, really what we would call vertical signaling inhibition, where if you have a pathway, you try to hit multiple nodes in the same pathway in a vertical manner. And that's an approach that's being pursued as well. And then the third area, which everyone talks about, but no one really knows what's going on, of course, is combining targeted therapies and immunotherapy. I'm a leader of a lung cancer Stand Up to Cancer Dream Team, and one of our goals is to figure out how to combine KRAS signaling inhibitors, MEK inhibitors, ERK inhibitors, with PD-1 checkpoint blockade in KRAS mutant lung cancers, trying to understand the effects of targeted therapy on the immune microenvironment and so forth, and how targeted therapies might affect the antigenicity of the cancer so that we might be able to combine those therapies. And those are trials, a lot of trials are now underway throughout the world combining immunotherapies and targeted therapies. And there's another word of caution there is that there's a lot of toxicity when we add these things together. So we probably need to figure out more about what's going on, what's contributing to that toxicity.
Thanks, Jeff. Jamie?
Good morning, everyone. I'm Jamie Friedman. I'm head of clinical development at Medimmune. Medimmune is the biologics arm of AstraZeneca. And I oversee oncology as well as other therapeutic areas. Oncology is the area of biggest growth within Medimmune, and that's driven by our interest in immuno-oncology agents. You've heard a lot about it over the last couple of days, and there are many classes of agents in addition to the checkpoint inhibitors. There's T-cell agonists, there's agents that target the innate immune response, and you can imagine that the number of potential combinations you can do in immuno-oncology are exponential. So there has to be a way of filtering out the best possible combinations. The specific question, Jens, that you asked was, what do you think is driving the proliferation of combinations? I think it's probably threefold. The first is, I think that there's always a tendency to try to get a better response in cancer patients over any individual agents. So the potential for getting better efficacy with the goal of cure in cancer is to combine agents because you can attack multiple pathways. I think that there's a lot of amazing scientific hypotheses that need to be tested, and the animal models are not sufficient to experimentally evaluate combinations, so it really needs to be done in patients. And that also relates to the first point that I made with the intent of improving efficacy. But there's also this behavior, I think, of company sponsors to do lots of combination trials where their particular molecule doesn't have activity and they're trying to find a role for that molecule. And I'm not sure that's really a good use of combination trials. The empiric combination of various agents without strong scientific hypotheses sometimes can work, but I think the failure rate is going to be very high, and it just consumes the number of patients that can be used for strong hypothesis-driven research. So I would make a plea that we shouldn't do as many combination trials, that we should be very diligent and rigorous around how we evaluate what combinations to be done, whether it's immunotherapy together or immunotherapy with target agents or chemotherapy, but that's, I think, what we need to try to achieve as a society.
David.
Good morning. I'm David Schenkein, honored to be here today. I'm a hematologist. I'm currently the CEO of a biotech company in Cambridge called Agios Pharmaceuticals. We're primarily focused in cancer metabolism, therapeutics, and as well as in rare genetic diseases. Before coming to Agios, I was the head of oncology and development at Genentech. My real exposure to combinations began as in my clinical career, in which I was a lymphoma doc and still am to some extent, and really got exposed to seeing the fact that we could cure large cell lymphoma with CHOP. And in 1986, when I was a first-year fellow, we saw the results of a large randomized trial of 5 different regimens, 5, 6, 7 drugs, none better than CHOP. But it was pretty clear that our ability to cure widely metastatic disease depended upon 4 or 5 drugs that had empirically been put together, all had single agent activity, but on their own cannot cure the disease. And we outright cure a significant fraction of patients with large cell lymphoma. And so, over the past, 25 years watching oncology development, I think similar to what Jamie said, I think it's been a little disappointing, particularly in the genomic era we're in now, that it still feels like most of the combination trials that I see at the meetings or on clinicaltrials.gov are largely empirically driven, as opposed to truly rigorously scientifically driven, either combining agents for convenience because a particular sponsor happens to have them in their portfolio and therefore it's easier to do, or for regulatory purposes because combining with standard of care is something that often is something that has to be done. So I think we're at a point now where our genomic understanding is at a level that we should be able to do much more scientifically driven combinations. I do think the future of curing widely metastatic tumors will still be largely combination-based therapy. And then finally, I'd say the other thing that's been a little disappointing over the last decade is how few of the new drugs are being improved in oncology using a companion diagnostic based on a true genetic marker. We've been talking a lot about it, but it's still the minority of drugs that actually have a pre-specified population of patients. And when we get to that point, I think we'll be better off, as Jeff mentioned, on combining drugs because they have a much more narrower profile. Their safety hopefully will allow us to do truly science-based combinations.
Good morning, I'm Bill Sellers. I'm head of oncology at NIBR, the Novartis Institutes for Biomedical Research, and I oversee early drug discovery and early clinical development for oncology. Prior to that, I was at the Broad Institute and with Ken at the Dana-Farber, running a cancer genetics program. I'll also echo some of the tensions, and I think in particular what David said about this desire to put combination therapy on a rational and hopefully preclinically motivated basis versus what I would agree was a very disappointing set of empirical trials, mostly either standard of care or just someone's best idea for the moment. And I would add one other aspect to that, which is also disappointing, which is we're still looking at incremental gain over single agent, or the addition of a new compound to a doublet is still incremental. And I have great skepticism that we will incrementally find a cure to a given cancer. In fact, I sort of in my mind have this concept of blocking standard of care therapies. That is, certain standard of cares get approved, and not only aren't they very effective, but they will actually prevent the cure of that cancer by forming a basis upon which people believe they have to add a drug. So gemcitabine in pancreas cancer may be an example where why that has to be a standard of care for a new combination would elude me. The second aspect is this desire to put combination therapy on a preclinical footing that's robust. And Jamie mentioned the models don't work, especially in IO. I'm not even sure there's a point in doing the IO models as they exist today. I do think targeted therapy at the preclinical side, when done at scale now, is beginning to unveil ways to think about coming up with combinations. And one way we've tried to simplify that problem is to conceptualize certain genetic forms of cancer as having a foundational genetic alteration, such as EGFR in lung cancer, which Jeff's been studying, and IDH mutations in either AML or glioma, which David and Agios have been studying. And to say that with that first therapeutic against that foundational genetic alteration, which we call the anchor therapeutic, whether it even exists today or not, there will be an anchor therapeutic against that. We want to find the next agent that will work with that to sort of simplify the problem to what's the second agent that goes together with the anchor therapeutic, whether that's an IO agent or a targeted agent. So this is one of our attempts to sort of move the preclinical science forward into informing more than empirical trials.
Thanks. So, I mean, I'm looking at this world as an investor, and I have to make decisions. And everybody knows right now that the theoretical number of combinations is mind-boggling. So I have to somehow figure out, I mean, what is the horse I'm betting on, and how do I make decisions? What combinations I, try to pursue, try to test, try to build a rationale for. I mean, how are you guys actually making decisions on a daily basis? I mean, you're looking at the world, there's a gazillion of things you could do. I mean, what are really the tools we have today to make decisions that really could lead to a quantum leap in some of the combination therapies? Ken, what do you want to say?
So one of my favorite concepts right now is Achilles' heels. And what I mean by that is that the genetics of all the diseases that we study are very complicated, and in fact, there's genetic evolution. There are drivers that underlie the primary biology of the various cancers, but they confer a phenotype, which is, in my view, in many cases, a vulnerability. So a couple of quick examples. In multiple myeloma, there's excess protein production. And a long time ago, the proteasome degradation of that protein was targeted. And a combination was predicated to block not only the proteasomal degradation, but the agrosomal degradation as well. That's the only way for protein to be degraded. So there was a vulnerability. More recently in myeloma, we have, as I already mentioned, lots of DNA damage, lots of ongoing constitutive DNA damage. And in fact, there is in certain subsets, amplification of MYC, for example, large amounts of replicative stress and increased ROS, or reactive oxygen species. So the combination that's going forward in the clinic now, predicated upon the genetic and then the phenotype that's conferred by the aberrant genetics, is to block the replicative response while combining it with an agent to increase the ROS. So the point is, I think in the preclinical targeted area, whether it be a targeted IDH or EGFR, or targeting sort of the biology that's the outcome of that aberrant genetics, I think it's very rational. I too share the skepticism that Bill expressed about the preclinical studies and what their utility is in terms of the models today for IO. I'm not sure we're really there yet in terms of modeling. But I want to stress one quick thing. I think the models can be useful not only for predicting targeted pathways that we may want to inhibit, but also to stop clinical trials from going forward. I'll just remember, I see David and I'm reminded of this, but when the proteasome inhibitors came out, there was a proposal to use proteasome inhibitors together with JNK inhibitors. There was actually a proposed trial. But in fact, they're absolutely antagonistic in preclinical models, and that trial never went forward. So I think the models, we always think of them as predictive of positive outcome, or at least a hypothesis that can be tested in the clinic. But I think, as I call it, a successful failure, a nice model that predicts that it should not translate, I would argue, is probably at least as useful.
So I think one of the decision makers is really, the biomarker diagnostic. And as you said, it is actually fairly disappointing how few new biomarkers and diagnostics are out there and are really used on a daily basis in practice. I mean, what's holding that back? I mean, we are sequencing the hell out of genomes now for quite some time and out of tumors. I mean, is there anything we need to do differently?
I mean, I don't know if I have the full answer. I think Bill and Jeff and maybe others can go deeper than this. I mean, I think that I've always thought there's two components to why we see so few new drugs approved in oncology today with companion diagnostic. One, it's the science is hard. It's really hard necessarily to find what is truly the predictive marker that predicts this patient is more or less likely to respond, unless it's an obvious genetic marker that is the hallmark of the disease. And obviously, CML and the Philadelphia chromosome was the poster child for that, or HER2 positive breast cancer. outside of a handful of examples, it's just really hard to find what that predictive marker is preclinically. I think the second thing is, and I think this falls somewhat onto industry, is they haven't been willing to set the bar high enough, and so haven't been willing to say, we're actually not going to take this molecule into the clinic until we figure out which are the right patients that are more likely to respond. You'll never get to 100%, but at least more likely to respond. And so I think it's a combination of that. But you know, Bill runs one of the biggest discovery shops in the world and deals with this every day.
Yes, I think one of the aspects of this is just the time, right? Okay, the human genome was completed, you know, more than a decade ago, but the cancer genome and its elucidation is a relatively recent phenomena, and the drugs that take advantage of the genetic definition of cancer still remain to be elaborated in some cases. So believe me, if you had a RAS inhibitor, you would have clinical trials with RAS mutant patients going on today. So I think there's a matter of time. There's obviously complexity, as David said, that also makes it difficult. I wanted to comment also on Jens, your question about how you would want to make decisions. And I think there are two aspects. Preclinical models haven't worked that great, but if you look at the way people evaluate combinations preclinically, you're lucky if people have done 2 xenografts or 3. There is simply no way that 3 experiments preclinically with 3 individual patient tumors can predict the outcome of a 50, 100, 200 patient clinical trial. So we've been trying to put the preclinical translational infrastructure on a more solid footing by building things like the cell line encyclopedia and this PTX, and this primary tumor-derived xenograft encyclopedia, where we're now running mini trials of combinations with 50 examples of pancreas and breast and lung. And I think some of that is starting to look quite interesting. For example, IGFR inhibitors, which failed in the clinic, look great in vitro, look terrible in the PTX model system. They completely abrogate the effect of MEK inhibitors. They don't synergize with MEK inhibitors. So we're starting to see some examples where if you do the translation at scale preclinically, it will work. The second is, it's still a matter of incremental. If we went for complete responses in the phase 1 trial of a combination, and you said, I'm not taking anything forward that doesn't have complete responses, almost nothing would go forward at the moment, and only the very best things would progress. So I think this is it's this always hope that you'll get rewarded by developing the combinations and get it registered versus really dramatic activity early on in a trial.
I think the idea of using diagnostics in targeted therapy is a completely testable hypothesis, even in phase 1. Where it becomes even more challenging is in the area of immunotherapy, where the preclinical models aren't predictive, and you know, you get into the clinic and you may have a hypothesis. For example, PD-L1 expression is a predictor for response to PD-1, PD-L1 targeting agents. But then as you are in the clinic longer, you learn more, and actually you may be able to find better diagnostics along the way. So for example, interferon gamma or CD8 infiltrates, or a combination of PD-L1 interferon gamma and CD8 infiltrates. And so I think the first approvals may come with one diagnostic, but over time, the diagnostics may evolve even after being approved for these agents, and that's how long it takes to figure it out. With regard to combinations, we discovered, as well as BMS, that if you combine CTLA-4 inhibitors and PD-1, PD-L1 inhibitors, you can have a synergistic effect in certain subpopulations. What we found and published in JCO was that very recently is that in the PD-L1 negative population of lung cancer, which is about 2/3 of non-small cell lung cancer, that you're able to see significantly better activity with the combination of CTLA-4 and PD-L1 than you do with PD-L1 alone. PD-L1 is active, very active in PD-L1 positive lung cancer, bladder cancer, head and neck cancer. But then when you combine with CTLA-4 inhibitors, you may be able to capture that population that's PD-L1 negative, and that's what we're going to be testing in our pivotal trials.
Jeff, I want to come back to resistance because, you know, for me, the challenge becomes even bigger if you think about resistance in combination therapy, right? I mean, if you had a single agent, you kind of can figure out, okay, that's kind of how the tumor can escape. Now you start combining different mechanisms, different point and signaling pathways. I mean, how much progress are we making in combination therapy to understand resistance? And are there any new tools that we actually have to develop to really understand how resistance should be looked at in a combination therapy setting?
So I have no idea, but I'll answer anyway. We don't really know that much about resistance to combinations. I mean, if you want to be honest, we know a decent amount about resistance to single agents, but we probably have a very superficial understanding of that process as well. I've been watching the Republican debate, so I know not to answer your question, but answer the question I want to answer. So I'm going to answer the question I want to answer, which is, it's really changed how we do research. I don't know the answers, but what we've started focusing on, I know Bill's group is also focused on, is not just trying to understand the cancer as it comes out at the end. So you treat and they go into remission for a couple of years, they become resistant, and then you figure out what the cancer is like at 2 years when it comes out. But what happened during that 2 years while the patient was on therapy? How did the resistance evolve? Did it come from cells that were there that had certain mutations that your combination didn't get to? Do cells undergo further evolution during those 2 years? So we've actually changed our research, instead of just understanding pre-treatment and resistance, to actually start to biopsy patients during the course of disease, to start to understand the biology of the cells that are still there, to understand if we can figure out ways to kill those cells in a proactive manner, building in combinations that you might not identify if you just look at the pre-treatment cancer and the resistant cancer. And this also goes for integrating immunotherapies and targeted therapies. Again, when we try to integrate those two things, we look at the immune microenvironment of the pre-treatment specimen. We have no idea how the targeted therapy is impacting the immune system and the immune microenvironment at the time of the targeted therapy engagement with the cancer. So you could have a cancer that's completely uninflamed. You now come in with the targeted therapy. There's a possibility that it actually induces cell death, it induces inflammation. People draw these cartoons, but no one has data for real. So I think this is incumbent upon both the academic and the biotech and pharma communities to start to interrogate cancers throughout the course of treatment more systematically, so we can understand these questions and maybe have more hypothesis-generated approaches of integrating both targeted and targeted, targeted immuno and immuno and immuno. Because if you're worried about having no biomarkers, wait till we have the immuno-immuno combinations, no biomarker.
So Jeff, just a question for you though, but if one of the things, as you well know, that requires, and everybody wants to do it, is multiple serial biopsies of a patient's tumor throughout their clinical course, throughout their clinical trial, which can be done maybe in a handful of institutions. But how do we raise that level of inquiry so that we can do that more routinely?
I think it's like everything else. So first of all, obviously we can also try to capitalize on non-invasive approaches, circulating DNA, circulating tumor cells and things like that. But I still think we need to get some tissue. And I think all you have to show is that when you do that, it makes a difference. You figure out what's going on, you develop a new therapy, and then everyone will start doing it. People said the same thing about genetics 7 years ago. We're never going to genetically profile every cancer. You show it makes a difference. You give big talks at ASCO and AACR. People say, geez, I want to do that too. And then they start doing it. So I just say, just start doing it.
Yeah, I will join the cheerleading of Jeff. I think that the idea of combination therapy expands not only predicated at the genetic abnormalities of the tumor that are going to evolve, but also the host. And I would make a very plea that we not only follow as we do patients over time, because that's really how we're going to determine mechanisms of response or mechanisms of resistance, but in many diseases we have sort of precursor conditions. We have in our area something called monoclonal gammopathy of undetermined significance that progresses to smoldering myeloma to active myeloma. And we and others are looking at not only the genetic events, but what are the host events, immune and otherwise, that might correlate. So I think the basis for understanding sensitivity or resistance, really, we can model it, we can try to model it, we can argue about whether the models are meaningful or not, but the real way we're going to determine this is by sequential monitoring of patient samples to see if there's common mechanisms of relapse, et cetera. I was just yesterday with this fellow, Greg Simon, who's in charge of the moonshot. And I know this meeting is all about big data in many ways, but the dream that we could actually have a database that transcends all cancers of genomically and otherwise profiled, maybe including immune profiling, of patients that could be followed over time, including all cancers, blood cancers and solid tumors, would be such a national treasure, because a lot of the questions that you're asking us up here for drug development, for combinations, for biomarkers, et cetera, would really be fast forwarded in that regard. So a plea for all of us to try to see if we can't unify. I know as a blood cancer doctor, I will share with you very big frustration in the sense that we find things in the blood cancer sometimes that we think are new that have been known in solid tumor settings for a long time, and vice versa. There's not enough of this cross-fertilization. But I think if we can, as a community, take all of this data, lots of it's already being collected in industry or in academia, and put it in a way that we can, maybe retrospectively, but certainly prospectively, collect this information so we can actually answer these questions based on data.
So, what I heard is that, I think there are some really new approaches in the preclinical settings where we can really do things fairly differently and get a lot of information. My next question as a venture guy, of course, is how do we translate all this into the clinic? Do you see that we need some fundamental changes how we look at the regulatory side of, how we run clinical trials in cancer?
Yeah, there's no question that, today, as Bill mentioned, for most agents, if you have a really active agent, and most of clinical drug development oncology for a brand new agent is done in the relapse refractory setting, and then the goal is to move it up front. And if there is a standard of care out there that actually benefits patients, actually leads to a fraction of patients who maybe are potentially cured, then it's very hard from a regulatory perspective to bypass that. And so you are, in a sense, somewhat boxed in from a regulatory perspective. It's not an absolute, depending on the scientific rationale, but you're almost boxed in to be able to either combine or compare with the standard of care. And it's going to take time. I think it's going to take a long time before that absolutely changes. And so, the goal we all have is to move as quickly as we can into the frontline setting, into the adjuvant setting. And if there's standard of care there, I think, there is almost a knee-jerk response to want to combine, and some of that is driven by the regulatory landscape.
The regulatory landscape, it seems to be evolving with immunotherapy. It's unprecedented to have agents that are in phase 1 trials and then advance very quickly to frontline combination trials, even in not combined with standard of care, but going against standard of care. And there's frontline trials of PD-L1, PD-1 inhibitors alone and in combination with CTLA-4 inhibitors in lung cancer, bladder cancer, head and neck cancer, and first line. So that really is unprecedented to be able to advance that quickly. I think the FDA realizing that, and the other regulatory agencies around the world seeing the dramatic benefit, the transformational benefit of these agents has really made them more flexible around how to be as aggressive as possible to get these combinations to patients as early as possible, hopefully with approvals. The breakthrough designation application, I think, helps with engagement with the FDA on a more frequent basis to work with the FDA to get these agents to the market. So I do see the regulatory agencies enabling combinations, particularly for ones that look very active in combination.
Although I would argue that in some of those diseases, that's because the standard of care is not effective.
Yeah.
And I think, just one comment on that. It's not likely that there are hundreds and hundreds of transformative combination therapies.
Yeah.
Right? There aren't hundreds of them. though we're trying hundreds and hundreds of combinations. So there's this tension between trying a lot of stuff and the fact that there are only going to be a few truly transformative combinations. So if you think about the preclinical data you would provide to the FDA to motivate, let's say, even a window phase 1 trial in a untreated or prior to standard of care, how often do you actually show them data that you've eradicated tumors in mice? Like almost never, right? So I think in a data-driven way, and Jamie was sort of speaking to this, when you have strong data, I think regulatory agencies can move. It's sort of incumbent on us to actually generate really strong motivational data saying we think we have something special that warrants this type of early accelerated trial in a non-standard of care treated population.
So Bill, in your experience putting combinations into the clinic, have you found that the agencies want to see from a safety perspective the combinations in preclinical tox studies, or can you just go directly in with
if it's neither agent has been in the clinic, you would need single agent exposure in humans. But we've not been asked for formal combination toxicology experiments. We have had experiences where a new agent was at sort of dose level 2 when a second agent that was still investigational was added. So we have had very early novel combinations start without formal toxicology of the combination. I actually think we're rethinking whether we need more investigative toxicology, to Jeff's point about schedule and routes of administration and intermittency, because I do think we've seen things even where spacing dosing has made retrospectively gigantic differences in toxicologic outcomes, which we could have identified preclinically had we done the experiments.
I would agree with Bill that the agencies have made a sea change over the last probably 7, 8, 9, 10 years on allowing novel combinations to go forward without as much preclinical safety data as we thought we'd need, depending on the mechanism. So that's obviously very encouraging to see that degree of novels allowed to go in, and it allows us, I think, to really try and find, is there truly a synthetic lethal pair that we can find between two agents or even more? Again, we are going to have to watch safety, as Jeff mentioned earlier.
Can I ask you a question? What do you do if you have one agent that you think is really effective? It's really effective as a single agent, and the PFS is 3 years from the single agent. But you have an idea, data, whatever, to suggest if you do this combination, it'll even be longer. That it would prevent resistance or it would deepen the response or whatever. Is there any easy trial to do to determine the benefit of the second drug that doesn't require a large randomized study? Are there any novel approaches? Do you know what I mean? Rather than having to go out 4 or 5 years to figure that out?
Well, not for the ultimate
yeah, but just for benefit, a good signal.
But even so I think ABL001, this new allosteric ABL inhibitor, is an example. It's going into a disease where PFS overall survival is 80% or higher at 10 years. But we believe the molecular response as measured by BCR-ABL transcript and the number of patients that will achieve complete molecular response, which even on first-line therapy is around 40%. If we said we wanted to double that, you would need like 20 patients to know that on the combination. So I think PFS doesn't mean complete response, right? So if you set your bar at eradicative therapy, then you'd have very small early signals.
Yeah, I would agree. I mean, I think it all comes down to finding surrogates.
Yep.
Surrogates that you think are likely to predict true clinical benefit. Now, the term likely is there is for an important reason, because they're not definitive. But unless they're, you're putting in patients into molecular CR, then it's pretty high likely. But I think there's a lot of opportunity to develop novel surrogates. Now, sometimes those novel surrogates will be used from a regulatory perspective, so you don't have to do a 15-year clinical trial. And others will just be for your own internal use to give you more confidence that this is the right trial or to continue that trial.
It's worth pointing out the second generation ABL inhibitors were approved on molecular response, even though at that moment in time, molecular response was not known or definitively proven in any randomized trial to be a known predictor of outcome. It was known retrospectively to be a predictor.
So I think what we do more often, Jeff, in answer to your question, is we have agents that have more transient responses, and can we extend that? That's an easier signal to look for. So I mean, a lot of the target agents like MEK, BRAF, they have responses in the order of a few months. But then the immunotherapy, what's remarkable about them is that you have this memory effect, and so you can have very durable responses. So the combination of that could give you an early signal if you see these very durable responses. And then I guess taken to an extreme, there are agents out there that don't have much single agent activity, but for example, hypomethylating agents in MDS, they work, but they don't have a lot of single agent activity. And then there are scientific hypotheses around induction of viral mimicry and the interferon pathway that would, leading to PD-L1 expression, that would suggest that if you combine a PD-L1 inhibitor with a hypomethylating agent, you actually may start seeing responses and durable responses in MDS and solid tumors even.
Yeah, I was just going to echo some of the other comments. I think CML is the paradigm, and they've been out in front of us for quite a long time. But the idea that we should set the bar high, so we should shoot for MRD. And I'll just share, in myeloma, we have PFS now routinely 5, 6, 7 years or even longer. And we're not going to be able to develop new agents together unless we have a surrogate marker. We're looking at minimal residual disease, but some parameter that we can look at 18 months that's going to predict for benefit at 10 years. And I think the FDA, I just wanted to sing their praises, several here already have, but the FDA has been extremely proactive at trying to help develop new strategies, new clinical trial strategies. Breakthrough and others have already been here. But again, part of this new Moonshot initiative is a big emphasis on new novel trial designs. And that's not only those of us in this room, but that really involves the FDA. There's what's called a virtual center, where the folks at the FDA are going to rotate through NIH and CMS and even academia and industry. They're going to be very, very well trained, but the idea is to do what we're all saying, to do much smaller, informed trials with hopefully surrogate endpoints that can give us an answer early on, which I think is extremely, it's absolutely critical for two reasons. One, if we're going to develop novel combinations in this panel together, we can't wait PFS or OS like we used to be able to. And secondly, for those of us who still see patients and in practice, we need to know how we can use, Bill knows in CML how to use, MRD. We don't know in many other areas. So we really need to work with the regulators to define how to measure a surrogate marker. Then you'll start to see more clinical trials. And then we can use it together to develop novel targeted agents or immune agents, and we can use it in practice in a much more informed way.
Well, I think those surrogates are going to be even more important when we're trying to do combinations, and when people have tried to do combinations where one of the drugs you're combining with has no intrinsic activity on their own.
Right.
And they're not having to wait for a long clinical trial. If both are active agents, then it's a little bit easier. But if it's not active at all, then surrogates are critically important.
Right.
So I just realized that we have mostly talked really about traditional chemotherapy, targeted approaches, and IO. What about radiation therapy? Does that play into this whole combination game as well? And how do we approach that?
Ken?
Ken?
Yeah, I'm the least qualified, I think, to talk about this, but I don't think we really know. I think the way I think about radiation therapy is a DNA damaging agent, and so honestly, it can be used in combinations, much as the way we think in the old days about DNA damaging agents. More recently, though, I think that to me, the most exciting concept is radiating patients with solid tumors and thereby, if you will, priming for an immune response, perhaps against neoantigens, but certainly against the autologous tumor. There are multiple protocols that I'm aware of and combinations, Jens, of radiation with the view that you can, in fact, then, if you will, prime or stimulate an immune response, often now in the setting of the checkpoint inhibitors. So to me, that's the most exciting part of the radiation now.
Yeah, I was going to add to that, that I think the combination of radiation and checkpoint inhibitors has really brought to the forefront that combinations with radiation therapy. I mean, radiation historically has been thought really as a local treatment, and then you add a systemic chemotherapy on top of it, particularly in lung cancer, head and neck cancer, there's other cancers like that. But I think what has happened with what Ken commented on, that you get an inflammatory response in the tumor, that will sensitize the tumor, the local tumor, to the checkpoint inhibitors, but also have an abscopal effect where you can have more distant benefit in metastatic disease. So not just the local treatment, but you might get a more broad effect on the patient.
Yeah, I just wanted to come back to the fact that it's really remarkable how quickly IO drugs really, found their way into patients. I think this is really interesting. But the flip side of that is, of course, that if you have toxicities that are based on aberrant immune responses, it's pretty scary. I mean, do we think we have a handle on kind of the toxicity risk in IO?
So I can comment first on that. I think the emergence of immune-related adverse events is not something that you see necessarily in the first 4 weeks of treatment. It's often these adverse effects occur after multiple cycles of therapy, and so the traditional drug development paradigm in phase 1 dose escalation, when you're looking at the DLT period for dose-limiting toxicity that's confined to 4 weeks, doesn't really apply anymore because you get these later toxicities. So that you need to redefine that window as being a longer window, and we're seeing that the regulatory agencies are actually imposing that on the phase 1 trial designs, that you look at 6 to 8 weeks, and then you still need to look at cumulative toxicity over time and put that together before going into phase 2 trials. In terms of managing them, I think now that we're seeing them more frequently, we're coming up with treatment algorithms for these immune-related adverse events that helps us manage those in the clinic. But some of them can be very severe and even life-threatening and somewhat idiosyncratic, unpredictable. It'd be great if we found markers that could predict who's going to get those toxicities and who doesn't.
Mm-hmm. Okay, so we're getting close to the end of our time, and I know that one discussion that will come back again and again and again, especially in the political season we are, is the whole pricing discussion. And so I think we also should bring it up in this panel, because I think combination therapies, as we have seen in antiviral compounds, for example, is a pretty interesting discussion when it comes to how you price these things. I mean, how do you guys think about pricing, especially on the company side? Is there an active thought process how you actually approach this?
Well, I mean, I think there's a lot of discussion around how to think about price, and I think it's most important to link the value that you're providing to patients to that equation. And when you start getting into combinations, that's starting to get complicated. But I think if we always go back to what true value are we providing to a patient, has it been, as was mentioned earlier, a slightly incremental improvement? Or has it been a game changer? And have we really changed the paradigm about the way we treat this disease? I think that should be reflective into the cost analysis. But as we think about stacking 2 or 3 or 4 new medicines together, I hope we lead to a cure if it's going to be that cost.
And how do you define the contribution of every drug? And then come up with, you get that much for that, and you get that much for that.
I was going to say, I totally agree with David. I mean, it depends on the magnitude of the effect of the combination, and you look at the contribution of components of the individual agents, and unless it's, completely transformative over monotherapy, you're not going to get what manufacturers hope to get, which is double the price of a monotherapy. I mean, that's what manufacturers want, because they argue that's the investment that they made into those 2 individual drugs that they combine, and they want to see the return on investment. But the payers aren't going to give you that reimbursement unless you show significantly better activity of the combination.
Yeah. It'll be interesting to see if the larger shops that have all the agents they need, or they're combining 3 or 4 from their own shop, are going to start bundling. We're not quite in that situation yet.
I mean, this might not be popular on the industry side, but if we were held to a higher standard of benefit,
Yeah.
we would not invest in weaker combination effect.
Right.
As a,
Okay, I think we'll have to end it here. I hope we gave you a little bit of an insight into the daily decisions we have to make. Thank you very much.
Thank you.